All right. Good evening, everyone. Welcome to the second series of the Native Plant Appreciation Month speaker series. I'm here tonight with Julie O'Donnell from the Native Plant Appreciation Month committee, who's worked hard all year to plan for this month of celebration. So thank you all for joining us, and I'll allow Julie to introduce and host our speakers. Thank you. I want one reminder, this program is being recorded. Um, and we'd like to have comments in the chat and questions in the Q&A, please. Thank you and enjoy the program.
Thank you for being here. I am also a member of the Central Puget Sound chapter, and in honor of Native Plant Appreciation Month 2025, I would like to welcome you to the second program in the series, Exploring Biodiversity, Exercises in Community Science. We invite you to learn about
Where's my phone?
learn what it is and how you can be involved. And we invite you to be part of the Washington Native Plant Society BioBlitz from April 20th to 30th. You will also find the webpage for each chapter includes activities to celebrate native plants through webinars, classes, field trips, plant sales, and volunteer opportunities. The mission of the Washington Native Plant Society is to promote the appreciation and conservation of Washington's native plants and their habitats through study, education, and advocacy. If you are already a member, we thank you. Membership supports native plant conservation and education, and it connects native plant enthusiasts across the state. To learn more about our work, please visit WNPS.org and consider becoming a member or leaving a donation. Your support is appreciated. As a member of the Central Puget Sound chapter, I would like to acknowledge that I am speaking from the lands of the Coast Salish people. They are the original and continuing stewards of the coastlines and forests of the Salish Sea. Before our speaker begins, I would like to invite you to attend next week's program on April 15th at 7 p.m. The topic next week will be rare native plants, and that's on Tuesday on Earth Day. So we hope you can join us. Be sure to pre-register to receive the Zoom link for that program. And also remember, there are two speakers tonight. So we'll listen to Barry Brosi first, and then we'll do Q&A and comments, and then it will be Regina Rochefort. So please stay with us for the whole program, and I'm sure we will learn quite a bit. We're looking forward to it. Thank you for being here. Barry, are you ready to
Yes,
kick it off?
absolutely. Yeah.
Okay, thank you.
All right. Well, thank you for that introduction. And I'm going to be talking about MeadowWatch today. So this is one of those community science programs, and I'll kick right off and get into the talk. So MeadowWatch is a citizen science program, and it's focused on really understanding how climate change is impacting the timing of wildflower reproduction in Mount Rainier National Park. And I'm going to unpack all of those terms in just a few more slides, but I wanted to give a very brief overview before going into anything else. And I also really wanted to start by acknowledging our amazing MeadowWatch team. And I'll begin with acknowledging Yannicka Hillary Slombers, who really is the founder of the MeadowWatch program. She used to be on the faculty, the biology faculty at University of Washington, where I'm also on the faculty. And she moved to ETH in Zurich in Switzerland in 2020, which is coincidentally the same time that I arrived at University of Washington after being on the faculty at Emory University in Atlanta for 10 years. Um, so it was really sad for me that Yannicka left UW, but it was also, an opportunity to really get involved with MeadowWatch since there was already a lot of alignment with my research program at that time. So fortuitous alignment, even though I'm sad that Yannicka is no longer here with us at UW, but she continues to remain very involved in the program. Another really, really important person for our program over the last several years, last 3 years, has been Minogia Chandar, who's my lab manager, and also really the volunteer coordinator for MeadowWatch. She's actually going to be going to graduate school in the fall, so she's going to be leaving us as well. But we're in the process of hiring a new lab manager. And the other person I really wanted to acknowledge is Madeline Straight, who's a PhD student in my lab, who's been really integral in bolstering MeadowWatch through her research program. And I'm going to talk about some of the cool work that she's doing down the road in the talk. All right, so in terms of an outline today, I'm going to talk about what we do and why with MeadowWatch. I'll get into some of the scientific results that we have, and then I'll talk about some next steps. So some stuff that we're really excited about, including a very imminent expansion of the MeadowWatch program to go beyond plants to also include pollinators. And then I'll end up with how people can get involved. So yeah, what do we do and why do we do it? I'm going to go back to that very first slide I showed, that we're a citizen science program. We're looking at climate change and phenology or timing of wildflower reproduction. So I'm going to unpack all of those terms now. So in terms of that first term in blue, we are a citizen science program, and the program tonight is on citizen or community science. And what that means is that volunteers, not professional scientists, I mean, some of them can be experts, but they don't have to be experts, are the ones who are collecting and or processing the data, and really doing that as part of an organized scientific project, part of an inquiry. So they're really this is where everyday people are really contributing to the data, which I think is really exciting, really fulfilling to be part of that. And then most of these citizen science programs also include some kind of outreach or educational component. Now, the second term I mentioned in that background slide is that we are focused on climate change. And I just want to say that there is really broad scientific consensus that climate change is occurring. Here in this slide, I'm showing some data on two facets of that. So first of all are some temperature data on the left, and we also have some snowpack data on the right. And these changes are happening globally, but they're also happening locally here in the PNW, and even more locally if we focus in at Mount Rainier National Park. So we really want to know what are the biological consequences of these changes in our climate. And we're focused on looking at how climate change impacts phenology or timing of particular life history events. So when plants and other organisms time, what they do is really critical for their survival and for the reproduction. So plants are using cues from the environment to do things like form leaf buds, flush the first set of leaves for the year. When those leaves start actually senescing, in the context of this picture, actually turning different colors when they drop their leaves. All of those things are really important in terms of maximizing the photosynthesis that a plant can achieve over the course of a growing season. And again, for MeadowWatch, what we're really focused on is plant reproduction. And I know I'm speaking to the Washington Native Plant Society, so probably all of you know this, but that's not true for all of the talks I give. So I'll just say that flowers and fruit are really key parts of the reproductive cycle of flowering plants. And, I think for a lot of people who aren't as invested in learning and understanding about plants, they see flowers, they think they're beautiful, but they don't understand that this is actually a really important part of the reproductive cycle. But really, that's part of the process that forms seeds, and then that in turn really forms the next generation of plants that we see. And again, like when I was talking about the timing of leafing, right? The timing of plant reproduction is really critical to the reproductive success of these plants and to their fitness overall. And there's two, I think, really important pieces of that timing. And one is that they have to have synchrony with other plants, right? So if you are the only plant of your species that's flowering at a particular time, there are no other plants that could donate pollen to your flowers to fertilize them, and your pollen can't also be transferred to other plants. So it's really important to have that synchrony with other plants, and it's also really important to have synchrony with other organisms, so with pollinators and with seed dispersers, so that those pollinators can actually visit your flowers, and later on, so that the seed dispersers can actually move your seeds around so that they're not all clustered right by the parent plant and all competing and potentially all getting diseases and whatnot. So those synchrony elements, the timing elements are really, really important. And plants use a range of environmental cues to time their reproductive cycle. When we're thinking about montane environments, so environments like Mount Rainier National Park, snowmelt is a really important factor when it comes to the cues that plants are using. And I'll talk about that in a few more slides. And interestingly, pollinators are using different cues and really have different constraints on their development. And with pollinators, what we're often really thinking about is the timing of when they emerge from overwintering, and often that's in the soil. It can be in a few other environments, but when those pollinators actually emerge and can begin foraging, and again, that's really driven by a different set of cues. And because there are differences in the cues plants and pollinators are using, that can lead to a potential for timing mismatches, which is something, again, I'll talk about in a few slides. But if we do see problems with timing, what that can end up with is reproductive declines, or in other words, when plants are producing fewer seeds than they might otherwise. And when that happens, and especially if that's happening over multiple years for these long-lived perennial plants that we tend to find again in montane environments, we can end up with really potentially very negative long-term consequences for plant populations. And another piece here is that there are many possible hypothetical scenarios of how climate change could impact different phases of reproduction and the timing of those different phases. And in MeadowWatch, we call those, and in the scientific literature on this more broadly, we call those phenophases. So the timing of different phases of the reproductive cycle. So when we were seeing flowering buds versus actual flowering, versus seeding, versus creating fruits versus releasing those seeds. So different climatic cues might be used. We could see a sort of spreading out of these events. We could see sort of relatively parallel shifts. And we really know very little about this. So there's a fair amount now that's known about flowering, but a lot less that's known about those other phases. And this is something that we do keep track of with MeadowWatch. So I think now I've unpacked all of those terms. So I'm going to show this slide one more time. So citizen science and climate change and phenology and wildflower reproduction. And now I'm going to put those pieces together, right? And one of the um, ways that we put the pieces together is to utilize citizen science and citizen scientists for this work, right? And there's a lot of science that's done, and in fact, the majority of science that's done is done with professional scientists or people who are training to become professional scientists, folks like graduate students and others. So it's a really important question of why are we doing this? Why are we actually using citizen scientists? And one really important reason in terms of phenology is that phenology happens really fast. So we need a high frequency of data collection to be able to understand when particular plants are moving from, say, the flowering phase to a fruiting phase, or from fruiting to releasing seeds. And that can happen, excuse me, over just the course of a day, or sometimes two. So we really want to get a very high frequency of data collection. And this is particularly true because we're studying multiple different plant species at Mount Rainier. And the second piece is that understanding the impacts of climate change is really important. And by enhancing public participation in science and the scientific process, I think we can really contribute to that. And also just even more than understanding climate change, right? This is understanding how science works, the importance of science for our society. And I think now more than ever, that's really critically important. So with MeadowWatch, we work with 16 focal plant species, again in Mount Rainier National Park. Those of you who are aficionados of high elevation plants in Washington State may recognize some or all of these plants. And we also work in two trail systems at Mount Rainier National Park in the Reflection Lakes Trail, which we have 11 plots that is in the Paradise area, so on the south side of the park near the Paradise Visitor Center. And in fact, if you look at the far side of the Reflection Lakes map there, you can see kind of the Paradise Visitor, Paradise Inn, and then a reference to the Visitor Center there. But going down Stevens Canyon Road just a little bit to those Reflection Lakes to get to the trailhead. And then we also have the Glacier Basin Trail, slightly longer trail where we have slightly more plots. This is on the east side of the park. So we have a little bit of difference between these two trails. Reflection Lake starts out a little higher, ends up a little bit higher, but is a shorter overall distance, again with fewer plots. And the Paradise area gets a fair amount of winter snow and summer precipitation, whereas Glacier Basin well also does. But being on the east side of the park, they're a little bit more in the rain shadow of Mount Rainier itself, so it is a drier habitat. Again, starts out lower, but we cover more ground on that trail. So that's really what we're doing is collecting data in these two trails, 28 plots, 16 species, and we have volunteers really out for essentially the entire span of the growing season of those 16 focal plant species. So now that's really kind of wraps up the what we do and the why parts. So I'm going to transition into some of our scientific results. So I'm just going to dive right into these. So I'm going to start out with a plot, and this is just from one of our species, subalpine lupin. And this is not even actually the full set of data that we have. So we did start collecting data for MeadowWatch in 2013. We've collected quite a few years actually of data, 13 years of data. This is really just the first 5 years of data that I'm showing in this plot, but when we add the more recent data to these data, the trends look very similar. So what we have here is the snowmelt date on the x-axis. So as we go along the horizontal, with later snowmelt happening as we go further to the right. So you can see from April, May, June, July, and then we have the peak either flowering or seeding happening as you go up on that y-axis, June, July, August, September. And what we can really see is that if we look at the purple dots, the flowering, the earlier snow melted, the earlier those plants bloomed, the earlier they seeded, and the later that date was, really the later those events happened. So we see really a linear relationship here, and that's actually also true of every other species in our data set. Although one thing that's interesting is that the slope of those relationships actually varies quite a bit with depending on the species. So the species are actually responding differently to the day of snowmelt. And let's see. Another key element that we see here is really reflected both in that last plot and in this plot, which is again that timing of snowmelt. And with the early snowmelt, what you can see is that not only are we shifting earlier, and this is the plot on the top, but that also these flowering and fruiting. So these data are just for flowering, but flowering, fruiting, other phenophases really gets spread out much more. Whereas when we have a later snowmelt, which is the more typical historical kind of condition, we see a much greater concentration or squishing together of those time periods when multiple plants are blooming together. One of the things that's interesting is that because those plants have different slopes of that relationship between snowmelt and flowering or fruiting, et cetera, we actually see different plants end up co-flowering with other plants in early snowmelt years relative to later snowmelt years. That's something that scientists really don't have a good handle on how important is that when we kind of shift the partners of kind of who is competing with whom for pollinators. So that's something my lab is actually really interested in delving into more, unpacking more. Another result that we found that I think is really interesting is that volunteer data is extremely high quality. So when we look at um, the estimate from a professional ecologist or training ecologist here, and we've had typically sent out someone 1 day per week over the course of the flowering season for MeadowWatch. And then we're able to compare their data with volunteer data. Really, they map along that one-to-one line that we would expect most of the time, which is really great. There's a couple of species that volunteers have a little bit more trouble with. Our Mertensia or bluebell, some of the Castilleja or paintbrushes, and volunteers tend to do much better with the flowering than they do with fruiting or seeding. And so, for those of you, for example, we're familiar with glacier lily flowers and fruits. It's the flowers are really obvious, and the fruits can be a lot harder to see. So that's not perhaps a surprise that some species and some phases are a little bit more difficult than others. When we look at flowering for the bulk of these species, we see, even higher than 91% accuracy for many of them, which is really great. Another really cool result I just want to mention here is that climate change may actually cause visitors to become mismatched with peak bloom. So this is a really cool analysis that Janneke and her lab did looking at some of the unusual snowmelt years, so early snowmelt years, and finding that basically the peak of visitation happens in Mount Rainier National Park, sort of irrespective of when the visitors are showing up. So visitors kind of come, they're not, they're like, oh, July is the time to see flowers, so they come in July, even if it was an early melt year. And I think another really neat thing that they did is actually analyze a lot of trip reports from the Washington Trails Association and specifically focusing on those trip reports from Mount Rainier National Park. And those really suggest that wildflowers drive visitor satisfaction or hiker satisfaction as much as bees do, right? So when visitors are going at the wrong time of year because of early snowmelt, they're going to be less satisfied with their trip than they would otherwise. Yeah, so there's really a ton that we can do with these data. And now I'm going to talk about some next steps, and some of that will be some fun analysis plans that we have underway for analyzing the data. And some of it's also an expansion that's going to happen to actually expand MeadowWatch to include pollinator monitoring. So I wanted to start with calling out again Madeline Straight, who's one of the PhD students in my lab, who's really just done an amazing job, made some massive contributions to MeadowWatch, but she's working on a project that I think is really cool, which is analyzing potential reproductive failure in plants in the MeadowWatch system. So if we see a plant was flowering in a plot, but then we never recorded fruiting or seed release from that same species, that could indicate that there was a potential reproductive failure. Similarly, if visitors say recorded that there were flower buds, but then never recorded flowers, again, we could be actually collecting data on some of these reproductive failures. And then we can link potentially those reproductive failures to early snowmelt years or other unusual climate years, including potential for, some lag effects. So if early snowmelt is stressing plants out, might we actually be seeing some of that impacting plants, for example, the following year? So I think a really cool analysis that Madeline is currently conducting. And one thing I'll say is some of these analyses, like this one, I think are really only possible basically now that we have 13 years of data. We'll have a 14th year after the summer, which is very exciting. But it does take some time, especially when we're looking at things like early snowmelt years. There are only so many of those in the time sequence that we have. So another thing that I am really excited about in terms of future directions is including pollinators. So my research program is primarily focused on plant-pollinator interactions and pollination. So this is very much right in the wheelhouse of what we do is an expansion to include pollinators. And right now, as I mentioned, MeadowWatch is currently only focused on plants. And so these timing mismatches between plants and pollinators that could be caused by climate change are potentially very, very important for plant pollination, for plant fitness, plant reproduction, the sustenance, the continued existence of many of these plant populations. But we have very, very few data on these timing mismatches, really only from a handful of places. They're often very focused on one plant species, and most of the data that are out there are really focused on one plant species that blooms really, really early in the season. So we have an opportunity to collect data on this in the same places as for MeadowWatch, where we know a lot about the phenology, the timing of the reproduction of these wildflowers, and really hoping to be able to do this over a longer time period so we can really inform this question. So in this coming summer, we're going to run a pilot field season for citizen-led pollinator data collection, which we're really excited about. This is made possible by a grant from the One Hive Foundation, which is a brand new foundation out of Vermont. And we're going to be using the same plants as our core MeadowWatch study. So again, that we'll have be collecting the data in the same place. In terms of our plans, we're going to be collecting observational data. So volunteers will not be actually capturing pollinators. But what that does mean is that the pollinator data will be relatively coarse in terms of the taxonomic categories. So it's limited to what volunteers can actually visually observe. Excuse me. And then the data collection will be focused on pollinator visitation to flowers. So not just seeing a pollinator flying around, right? We want to actually record a particular pollinator visiting a plant species. So we have data on both of those elements, right? We have data on the plant, but we also have data on the pollinator, and this will again really enrich our ability to look at timing mismatches and these things like competition, right? If we have two plants that in historically really never overlapped in terms of their bloom, and now they're having to compete for pollinators, and we're really only seeing the pollinators visit one of those two species, that's really important data to have, which is why we're really focused on the flower visitation piece. So we're doing this this summer with a pretty focused cohort of volunteers. So we have um, some volunteers who are really experts in pollinator ID or insect ID. And then we also have a set of MeadowWatch volunteers who've been working in the MeadowWatch program for quite some time, but who really don't have any pollinator experience. And so we've really set that up to be able to kind of inform both ends of the spectrum in terms of that volunteer experience. And we're, as I say in the slide here, we're really excited to see how that goes. And we're really hoping that we can open this up to a really broad range of volunteers to collect pollinator data in 2026. And that's a great segue to the next topic I want to cover, which is how you can get involved. And I want to say, first of all, a huge thank you to all of our Washington Native Plant Society volunteers. So when Yannicka started MeadowWatch, some of you may have even been there, but she gave a talk to Washington Native Plant Society. And that talk really got the first core set of volunteers for MeadowWatch that I believe started collecting data in 2014. Ionica had some folks from her lab, I believe, that were collecting data without a sort of citizen science component in 2013. But either way, really, Washington Native Plant Society has been sort of foundational, crucial in making this work happen. If this is something that you haven't done in the past and you're interested to do, I really encourage you to, or if you're even potentially interested in doing it, I'd encourage you to sign up for the MeadowWatch listserv. That's at www.meadowwatch.org. And we will have volunteer trainings in June in both Seattle and then down in Ashford, so near Mount Rainier National Park. And I wanted to just mention some of the benefits of volunteering. So again, it's a really, I think, exciting opportunity to be able to contribute to science and to contribute to science directly related to climate change at a time when that science really is under attack and where it's really unclear what the future of climate change science, at least federally funded science, is going to be over the near term. So I think it's something that you can actually do to combat that problem. And a couple of maybe more directly tangible benefits is that or that you get free entry to Mount Rainier National Park. Last year, they set up a timed entry system where you had to have a reservation. You could only get in within a certain window. By volunteering, you can actually bypass that timed entry. We don't know if timed entry is going to happen again this year with some cuts in National Park Service staff. But if that does happen again, with MeadowWatch, you'll be able to bypass that timed entry when you go in to volunteer to collect data, of course, only. Another really nice element is that many of the campgrounds in Mount Rainier National Park fill up months and months in advance. You can camp for free for 2 nights if you're collecting data um, as part of MeadowWatch, and you need to kind of plan that about a week in advance. But again, a really nice benefit. And I'll also just add that in terms of the volunteering, where we really need the most help is going on weekdays. We get tons of people interested in weekend volunteering. That's also when the park is obviously most crowded. But we love to have volunteers going in on weekdays, and we also really love it when volunteers are contributing to data collection early and late in the season. So they may be missing the absolute peak bloom of, beautiful wildflowers. But it's really important that we get that data early on and also later on in the season. So with that, I'm gonna wrap up. Thank you so much for attending today and for your attention, and I'm happy to take any questions.
I am looking for some questions. The chat looks, you can put your questions in Q&A and no open questions. Okay. So, thank you for that, Barry. That was wonderful.
Yeah.
I just your photographs and thinking about being in the mountains is so, so appealing. If I didn't live north of Seattle, I'd really consider
Yeah.
joining you. It would be just a great way to practice your botany skills. And
Yeah.
anyway, it
Yeah.
looks like a really wonderful way to be out, help community science, and learn. It's the best way to learn too about the different species of both plants and pollinators. So
Yeah.
yeah. So, okay, I still don't see any questions. I think maybe we're patiently waiting for Regina's program, the second part.
Oh yeah, looks like we got a question here in
Oh,
the
there's a
Q&A.
Q&A. Okay,
Yeah.
right. Yeah,
So
I'm
yeah,
glad.
I mentioned this yeah, really briefly, and I can actually pop back to that slide. So we have two different trails that so the question was how many miles do volunteers need to walk? So we have two different trails, and so these are relatively long and relatively strenuous hikes, so they do go up um, a fair um, a fair amount of elevation, right? So we are looking at a 7 mile trip for Glacier Basin and 5.5 miles for Reflection Lakes. So you need to be in decent shape, but we do have how fast or slow you take it. It really is kind of up to you. For many of the volunteers, when they do one of these hikes, it does take them the better part of one day. So hope that gets to answering that question well enough. I'll get into that second question, which is for our pollinator work, will we be looking for caterpillars of potential pollinators? No. We're really only looking at that flower visitation element, but that may be something I don't actually know the answer. You may want to ask that for the next talk. So ask that to Regina, who's really doing this butterfly work. And we have a question saying, I've participated in MeadowWatch for the past 6 years, and it's a great program. So first of all, thank you so much for volunteering. I really appreciate it. And can you talk about which data you're actually collecting so members know what's expected? Yeah, so we are collecting data on, again, several different phenophases. So when volunteers show up to one of the plots, and I'll actually change the slide here. To show avalanche lily, and basically we're looking for data on, are there flower buds? Yes or no? Are we seeing actual flowers? Yes or no? Are we seeing fruits? And then are we seeing release of seeds from those fruits? And again, we're doing that for 16 different species, but it's important to know that at each plot we really typically only have somewhere between you know maybe two and four species typically that you're collecting data on in a given plot. So I hope that covers that question. And then we've got a question. In my experience, do pollinators disappear before or after endangered plants? Um, I think it really depends, on the context that we're dealing with, and certainly we have seen um, both of those directions happen, right? Where the plant disappears first and the pollinator disappears first, and it really depends kind of on the the context in terms of drivers of those local or global extinctions. Yeah. And thank you. The last point there is the hike distance, yes, is in round trip, right? So we're not talking about a 14-mile round trip. Yeah. Um, yeah, I hope that covers those questions. I'm happy to take any other questions that folks have.
The question from Tina Miller. I've participated in MeadowWatch for the past 6 years. It's a great program. Can you talk about which data you are actually collecting so members know what is expected?
Yeah, I think I answered that. I tried to answer that in terms of collecting the data on the different phenophases and for those species. So I hope I got a clear enough answer. Did you have additional follow-up on that, Julie, or is that close
No, I'm
enough?
just
Okay.
reading the
Yeah.
question, the Q&A.
Yeah. Yeah. So I think I covered everything there in the Q&A.
Okay.
Wonderful.
Yeah, thank you. Oh,
Oh, and
wonderful.
then, yeah, one last question, which is how many hikes do you have to commit to, right? So, when you do the training, we typically ask that volunteers do at least two hikes. You know, in the end, we can't force anyone to do anything. So we certainly have some volunteers that will, just be able to do one. And sometimes, there are things like, well, I signed up for one and I did it, and then I sprained my ankle, and we're not going to come after anyone if that happens. We do find it really helpful if folks can commit to doing at least two rounds of data collection. And we have some volunteers that have been collecting data, almost weekly or in some cases even weekly, that really like to get out on those trails. They know the trails like the back of their hand, and really like to see and feel and understand those seasonal changes. So that's really helpful to us when we have folks that are coming sort of repeatedly throughout the season. But yeah, really our baseline request is two hikes. But I'd say, if you're not sure you can make that, we'd rather actually have more volunteers. We'd rather have more people sign up, come to the training. Even if you get one hike in, every little bit of data collection really does help us. Yeah, thank you for that.
Wonderful. Thank you for sharing with us and giving us some insight into the work you're doing. It is really fascinating to learn about it and hear about your results and perhaps more results in the future too.
Yeah.
Thank you,
And
Barry.
yeah, and just one last question came in, which is I'd love a CC of the species. Um, we do have these actually on our MeadowWatch website, so that's probably the best thing to do is meadowwatch.org, www.meadowwatch.org, and I can actually put that back up for you as well. That's up there on the top right, and yeah, we have a list of all of those species there. All right. Yeah. And I just thank you again for the invitation, and thanks again to all of you for listening and for your attention. I really appreciate it.
Right. And with that, we are going to switch to our next speaker. Regina Rochefort is with us, and she will be able to answer a few of those questions as well, especially about caterpillars. So I'm going to read your bio first, Regina.
Okay.
Do you want to get your screen up, do you think?
Yes.
We
Yeah.
had it up earlier, so.
Thanks.
Okay, there we go. Dr. Regina Rochefort established the Cascade Butterfly Project in 2011 when she was the science advisor at North Cascades National Service Complex. Prior to working at North Cascades, Dr. Rochefort was the botanist at Mount Rainier National Park and Everglades National Park, and also a fire ecologist at Big Cypress National Preserve. She received her PhD from the University of Washington, MFS from Yale University, and BS from Northeastern University. She retired from the National Park Service in 2018 after 40 years and now volunteers on the Cascade Butterfly Project and the Science Committee at the Dennis Conservation Land Trust on Cape Cod. We welcome Regina Rochefort. Thank you so for being with us tonight, Regina.
Well, thanks, Julie. It's great to be here, and it's really nice to follow MeadowWatch since we are pretty similar projects.
Okay.
See.
Make that as full screen as possible.
Yep. Okay. So the Cascade Butterfly Project is a community science project. And we designed it to track the effects of climate change on subalpine ecosystems, to communicate the effects of climate change to the public. And we also hoped it would supplement the NPS long-term monitoring program. We started doing baseline studies to get ready for the project in 2008 to about 2010. And then our first surveys were conducted in 2011, and we've continued each summer since then. Now, just as a little bit of background, I did mention that we wanted this to supplement the NPS long-term monitoring program. So if you're not familiar with this, I thought I'd give you a very brief overview. In 1998, Congress mandated that the National Park Service should monitor natural resources, be able to document the health of these resources. There were two stages, inventory and monitoring. And it was great that, as well as mandating us to do this long-term monitoring, there was also funding provided. But of course, funding is finite and costs go up. So the Park Service had to determine how best to structure this project so we could continue it in the long term. And they decided to group parks that had significant natural resources into networks. There are 32 networks nationwide. And in the Pacific Northwest, we're part of the North Coast and Cascades Network. So on the screen on the right, you can see there are 7 parks in the North Coast and Cascades Network, or NCCN. Starting with the Canadian border, there's North Cascades National Park Service Complex. As you head south, San Juan Island National Historical Park, Edie's Landing National Historical Reserve, Olympic National Park, Mount Rainier National Park, Lewis and Clark National Historical Park, and Fort Vancouver National Historic Site. So all the ecologists and physical scientists from the parks got together in about 1999 or 2000, and we had to determine what would we monitor for this long-term monitoring program. And the indicators were called vital signs. And of course, being ecologists, we wanted to monitor everything, but we knew we had to limit ourselves. So we first decided that we needed an overarching goal, and we would select climate change as our overarching goal. There was a lot of research coming out and being published around 2000, talking about projected changes in climate and effects that people were already seeing. So we selected vital signs, often different vital signs that could be monitored in all 7 parks, or at least a majority of them. And some of the vital signs we selected were glaciers, land birds, subalpine vegetation, whitebark pine, intertidal communities. But throughout our discussions, insects kept coming up because we're already there was already research being published talking about the declines in insects and in particular pollinators. But on our group, none of us were entomologists. So we had a really hard time determining what is the research question we could ask that we could then develop monitoring standards and analyze the data. So in the end, in 2000, we did not select pollinators, but we kept talking about it. And in about 2005, I became aware of the Rocky Mountain National Park Butterfly Monitoring Program. And this was a volunteer program set up by Rich Bray and Dr. Paul Oppler, and a couple of wildlife biologists, Mason Reed at Mount Rainier and Bob Kuntz at North Cascades. And I thought, this would be something we could do. We thought butterfly monitoring would be popular. We could get people to volunteer. And we also thought that butterflies could be monitored annually. And we would be supplementing the long-term monitoring program by seeing the steps annually that as ecosystems were changing. So the slide that you see before you is a sunrise meadow, which you may recognize. And many of the indicators that we were monitoring for long-term monitoring, we could only afford to monitor them every 5 years, or maybe 10. In the case of tree establishment in subalpine meadows, that's probably very appropriate because it takes at least 3 years for a tree to survive from seedling stage to maybe making it long-term. The tree in the foreground here is probably 100 years old. On the right side of your slide, you can see 2 histograms. The top is sunrise and the bottom is paradise. And it shows the number of trees that established in each 5-year period. So you can see in sunrise, not as many trees were coming in. That side, as Barry mentioned, is drier and hotter. Drought kills a lot of those trees that come in. So trees only established during cool, wet years. Paradise is a more benevolent habitat. As long as the snow melts early, trees can have a long enough growing season. So we saw trees almost every year. And often these trees established in years when glaciers were receding. So you can see looking every 5 years is probably fine for trees. But you wonder, what's happening to the plants in between? What's happening to the pollinators or to the insects? So that was a reason that we thought pollinators would be helpful. There were a lot of recent studies documenting pollinator declines. Dr. Art Shapiro in California had been showing that pollinators, butterflies, were moving up in elevation with warming climates. In Europe, I think the butterfly studies were going on. They've been several decades showing different species moving north with warming climates. So we knew that we would probably be able to document change and we should get out there. So by selecting butterflies, we would be picking a species that we thought was easy to learn. We could compare it to other studies. And although butterflies are not the most important pollinator in the subalpine, that probably is syrphid flies, ants, and bees, butterflies do reflect the same changes that are going on in those insect communities. And I guess to go on, we also thought that with pollinators, we'd be able to see short-term weather changes and long-term climate change, or the effects of those climate changes on butterfly populations. So I guess I have gone over why we selected butterflies. We selected them because they're easy to learn. There were a lot of people out there that already knew butterflies, and we also thought we would be able to attract volunteers. We could track changes, compare changes to other studies across the world. We started in 2011, and in 2012, we added plant flowering patterns to this. And of course, we picked plants because plants and butterflies depend on one another. And there were a lot of other plant studies. The National Phenology Network was getting started, and that would give us another comparison. So we had both short and long-term objectives. As I mentioned, we did some background literature searches and field studies from 2008 to 2010. At that time, we could find records for butterflies in Mount Rainier National Park, but most of them were more like iNaturalist, that they were one time seeing butterflies. It wasn't watching over time every week what would happen to a population. But in the Northern Cascades, we found very few records. So we knew that we really needed to improve our butterfly inventory for both parks. We wanted to track annual patterns of abundance. And another goal was to provide field experience to students. From 2011 through 2018, we were able to leverage with a lot of programs that were started by the National Park Service or USGS, where students could come out and work as interns with us. And that was important because sometimes when students are in college, they don't hear about what field biologists do. So we wanted them to see what we were doing. So perhaps they would come and work with us or other places that hire field biologists or naturalists. And then, of course, we thought we could communicate our findings. A lot of people don't go into a visitor center. And when we're out there, since we have butterfly nets and other people aren't allowed to carry them, we attract a lot of attention. So it gives us a great opportunity to tell people what we're doing and why we're doing. In the long term, we were hoping we'd see the effects of climate on butterfly populations. Not that we wanted to see effects or bad effects, but we felt in the long term we would be able to document trends, population trends, species richness, abundance, as well as flight time. Butterflies peak at different times, just as plants do. We wanted to see if those peak flowering or flight times were shifting, and were the plant flowering times shifting at the same rate. And Barry brought this up about plants and animals, whether they're changing at the same rate. Okay, so we have two types of monitoring, qualitative and quantitative. So the qualitative monitoring is just something that anybody can do. You don't have to sign up. You can go out and take photos and add to our inventories. As I'll show you, we only have 10 study sites, and across 6 million acres, our study area. That means we're not getting out to a lot of places. We may be missing a lot of species. So that's where the qualitative inventories help us. And then quantitative inventories that tell us about the relative abundance. And I'll go over that in a little bit more detail. So as far as qualitative surveys, all you really need is to hike and a camera and get out there. And the three platforms that I'm most familiar with are Butterflies and Moths of North America, iNaturalist, and to a lesser extent, eButterfly. But scientists use this data all the time, so it's really important to get if you can contribute to this. I think one thing I like about Butterflies and Moths of North America is when you upload your photos, there's an expert who will check your photos. So you don't have to know what you're seeing, but somebody will review it, and then they'll post your observation with your photo and a map showing you what you did see. iNaturalist is the community telling you what you saw or confirming what you think you saw. And sometimes people look at yours and sometimes they don't, but both are great avenues. So as far as our quantitative surveys, we originally had 10 study sites. We wanted all study sites to be in the subalpine. We wanted them to be a short hike. So our longest hike is 4 miles one way to any study site, but we really wanted our volunteers to be able to get out, get to a study site and back in one day. So this map shows our study sites. Starting at the north, it's Skyline Divide and Sauk Mountain in Mount Baker Snoqualmie National Forest, Cascade Pass and Easy Pass in North Cascades National Park, Maple Pass in Okanagan-Wenatchee National Forest, and then 5 sites in Mount Rainier, going clockwise: Spray Park, Skyscraper Mountain, Sunrise Rim, Natchez Loop, and Mazama Ridge. In the Northern Cascades, of course, we wanted to look across ecosystems, but there are fewer sites that are close, fewer trails, fewer roads. So that's why our study sites are spread out and spread between the national forests and North Cascades National Park. Unfortunately, we haven't been able to survey Skyline Divide for about 3 years since the road washed out and has not been rebuilt. And it would be, I think, about a 10-mile hike if you did try to get into it up a road without, with its current status. Cascade Pass, Easy Pass, and Maple Pass also have had spotty surveys due to fires. Sometimes they're closed due to fires, or smoke makes it unhealthy, but we can still get in there at times, and we're still hoping to survey in those areas. So as far as our methods, I think I mentioned all our study sites are along trails because we know we're in sensitive habitat. Each of our routes is 1 kilometer in length. We use the pollard walk method, which is a relative abundance. So on this slide, you can see Eddie on the right at Easy Pass, and he's walking with a butterfly net. He has a partner who's behind him, an observer who will note take. As he walks, he has an imaginary 5 by 5 by 5 meter box in front of him, 2.5 meters to the right, 2.5 to the left, 5 meters in front, and 5 meters from the ground up. Any butterfly that flies through that box or is sitting on vegetation in that box is identified to species. That's our goal, to get to species. Sometimes you have to catch it. We put it in a jar and look especially at the ventral side to see the marks by which we can identify the species. It's great to have at least 2 people when you're out there because it's really hard to walk and take notes and track the butterflies by yourself. It's also great to have at least 3 people or 4 because often people talk to us, and it's really nice to have one person who can step aside and explain what we're doing and even show the butterfly if we have it in a jar. It's also, sometimes you need a backup netter. Although Eddie, being the observer, was going first, and he determines what butterflies we're going to count in our tallies, one might fly past him and somebody behind him may have to catch it and identify it. We also do plant phenology using phenophases, as Barry mentioned, and ours are a little, we only have 4 phenophases, either not there, vegetative. This is a Lupinus latifolius, you can see in the bottom that's just vegetative. In the middle, it's flowering, and on the right, it's in fruit. Each survey route has a select list of plants, anywhere from 10 to 12 plants that are monitored. These plants were selected for one of several reasons. We tried to pick plants that were easy to identify. Sometimes a plant was selected because it's very characteristic of early or late season. For instance, we also had avalanche lily like MeadowWatch, but we're generally there after they're in flower. So sometimes we miss those, so we took it off our list. Or it might be because a plant is important as a host plant, such as Dicentra, which is a host plant for Clodius parnassian. So as far as some of our results, right now we have 56 species on our inventories, 47 of which we've documented in North Cascades, or the Northern Cascades, the 3 areas, 46 in Mount Rainier, and there are 40 that are in common. As far as species distributions, I think we have a pretty good idea now of which species are widespread, which ones are occasionally seen, and which roots are a little different than other roots. And we're also starting to see changes in timing of peak abundance of butterflies and plant flowering. So of our 56 species, I'm just going to show you our most abundant. So these are the 8 most abundant species. Anna's blue on the top left is the most abundant across all our sites. It occurs on all sites. Silvery blue is next to it. Waterval's blue. And then Mariposa copper, which is also in the same family. Going across the bottom, Mormon frit, Hydaspes frit. These are both greater fritillaries, which are characterized by these large orbs on the ventral side, Arctic fritillary, and Western meadow fritillary, which are both lesser fritillaries. So I have ventral sides on all of these because although some butterflies you can tell just by looking at the dorsal side, like for instance, a Western tiger swallowtail butterfly, but a lot of them, like the blues especially, you really have to look at the ventral side to look for some of these marks, like are there orange arrows on the outer side? What's the shape of the dots on the wings? Are they on the fore and the hind wings? You can see a difference here between the silvery blue and the Waterval's, where the silvery blue, these dots on the hind wing are all similar shape, whereas on the Waterval's, you can see a big difference between the forewing and the hind wing. So I mentioned some of the other information that we're gathering that we're feeling pretty good about knowing what's out there is abundance. So thinking about Anna's blue, and on this photo you can see female dorsal side, male dorsal, and then mating blues and a ventral side. Anna's blue, as I mentioned, is the #1 butterfly, or the most abundant butterfly across all roots. It is the most abundant on Sauk, Easy, Maple, Natchez Loop, and Mazama Ridge. On this map, the size of the circle indicates the percent of the butterfly that we can see across all sites. So the largest circle is on Sauk, because Sauk and Easy Pass, because that's where we've seen the most butterflies of Anna's blue, most abundant. In recent years, though, this map was made in 2018. So right now, it would look a little bit different if I was making it today, because the last several years, we've seen quite a few on Mazama Ridge. So Mazama down here in Mount Rainier would probably have quite a few, have a larger circle if we were making this graph then. Another one is Western meadow fritillary. This is very abundant on Sauk Mountain early season. If you're walking, sometimes when you're walking up the trail, you might see 40 as you're doing the survey route. And I should say, it takes us anywhere from maybe half an hour to an hour and a half to do our survey routes. You walk what's called a meadow, a wedding march. So it's 1 foot together, 1 foot together. So you're going slow and looking all around you as to what butterflies you see. But sometimes, if there are a lot, it could take you an hour and a half to 2 hours to do the survey route. So it's Western meadow is most abundant on Sauk. On most of the other routes, it's very low abundance. And this is one, I don't know if any of you have seen the recent article that Colin Edwards is the lead author on rapid butterfly declines across the United States during the 21st century. So this was a large group of scientists using many surveys from across the US, and they looked at butterfly abundance between 2000 and 2020 and found that over this 20-year period, they found about a 20% decline in butterfly abundance. They also looked at regions, and the Pacific Northwest had a much lower decline rate. Over the 20 years, only 2% decline. So, and then they looked at species both across the US and in the Pacific Northwest. So Western meadow fritillary was one that was declining across the US and in the Pacific Northwest. Mariposa copper is another in the lichanid family. It's observed on all 10 roots. This is a butterfly that, if you get this image in your mind, you can often identify this one without catching it, especially if it is sitting on a plant, because you can see on this ventral side that the forewing and the hind wing are two completely different colors. So it's pretty easy to notice. And this is very abundant on Cascade Pass, Easy Pass, Sauk, Mazama, and Natchez Loop. This is one that Edwards lists as increasing both in the Pacific Northwest and across the US. Now, thinking of some of our less abundant species, purplish copper is only on 8 roots. And although it's on 8 roots, I list it as lower abundance because there are very few that we see when we see them. We don't see 100 at a time. This is one that Edwards also lists as declining, and we have not seen it on Spray Park or Sunrise Rim. Every year we do see new species or different species on a new species to a new route. So they may be out there, we just haven't found them. And I don't know if I mentioned, but our goal is really to survey each route at least once per week between snowmelt and the first frost. So we're generally out there early July through Labor Day, or sometimes till the end of September. And it's really great if we can have 2 or 3 surveys in the same week. Coronis fritillary is really very interesting. I don't know, it's on 5 roots, all the Mount Rainier roots. It was first observed in 2016 by Melanie Weiss on Natchez Loop. And since that time, we've seen quite a few of them, especially on Natchez and Sunrise Rim. It is a greater frit, and it's in the top 2 pictures here are the Coronis frit. The bottom 2 are Mormon and Hydaspes. You can tell the difference because one thing is Mormon come out earlier in the year. Usually the first time we're out on Sunrise Rim, they're flying. They also, the orbs on the ventral side of the hind wing are slightly different shape, and the coloring can be different, whereas Hydaspes is very reddish brown. Mormon and Coronis look more similar. But I think the most interesting thing about the Coronis fritillary is that it actually overwinters on the east side in the sagebrush. That's where it mates, that's where it lays its eggs. It mates there maybe around May. Then they move up to higher elevations. And that's when we see them, mostly females, but some males, coming up and flying around Sunrise Rim, Skyscraper. And we've seen them even on Spray Park, which is a little farther west, and recently at Mazama Ridge. And the females stay up there for most of the season, and then they head back down to the sagebrush and lay their eggs. And their host plant is Viola trinervata, which is in the sagebrush. So I listed an article here that David James and Jonathan Pelham wrote in 2011, and it's a great article, talks about the natural history and also shows migration routes from sagebrush up in elevation. And I feel really great that we actually have picked up not just butterflies that are hanging around the whole time in the meadows, but ones that are migrating and passing through. So as far as butterfly flight times and peak abundance, we do feel like we've been able to track this and see differences from year to year. So this graph extends on the x-axis from June 4th to September 24th. Each different color represents a different year, and it's all of our routes and all the butterflies, those that are identified as species, as well as those that are just identified as, say, a blue or a fritillary. But what I think is, and each point on the line is a survey. I think that what's really cool on this is you can see different years had different timing of peaks and different levels of peaks. So 2015, this is a year that Barry also mentioned, was a very early snowmelt year. Snow melted about 3 weeks early. We even saw gentians flowering up on Mazama Ridge in the first week of July. So not only did snow melt early, butterflies came out early and plants flowered early. 2018 is interesting because it's a peak abundance year in a more typical time, grouping all butterflies, the third week of August. But Leslie Reese at Georgetown, who hosts the database that we put our butterfly data in, said from all over the US, she was hearing that people were seeing high abundances of butterflies in 2018. Since we're all at slightly different weather or elevations, it's sort of curious, and I don't know why that is, but I think it's very interesting. 2024, even though we had low snowpack, if you remember, we had cool, wet spring, and the snow didn't melt that early, so we had later peaks in butterflies. Now, we also can see differences among species. Some react to the drier winter or lower snowpack or temperature. We're trying to sort this out. What are the cues they're each reacting to? In this slide on the left, it's Anna's blue, all roots put together. On the right side is the Western meadow fritillary. You can see both species did come out early in 2015. And they both had peaks in 2021 and 2018. I think the difference between the two is Western meadow fritillary is always comes out earlier than Anna's blue. And you can see that even on the 2024 later snowmelt year, its peak was still early. So this is an early season butterfly that does show some of the same reaction as, say, Anna's blue, which generally has peaks more mid-August, whereas at least on sock, Western meadow fritillary is early July, but they both reacted to 2015 and they both spread out their abundance, although Western meadow fritillary in 2024 was still fairly early in the season. Then we also are looking across sites. So this is Anna's blue. On the left, it's at Sauk Mountain. Sauk Mountain is a south aspect, melts out pretty early, only 5,200 feet in elevation. On the right is Mazama Ridge, which is at about 5,700 feet in elevation. And it's one of our latest sites to melt out because even though, and the Mazama Ridge route overlaps with the Reflection Lakes route partially for MeadowWatch. And if you're familiar with that route, if you go up Fourth Crossing to get to the Skyline Trail, although it's on a ridge, the trail is on a concave surface. So snow hangs there a lot later in the season. So though it's a higher elevation, it keeps the snow there. And you can see that the Anna's blue peaks much later on Mazama Ridge than Sauk Mountain. This gray peak here is 2021, which Ridge was about July 26th. The same on Mazama's Ridge was about August 24th. So we are interested in seeing if we're seeing overall changes in abundance over time. This is all of our data, just butterflies identified as species, going from 2011 to 2024. The arrow on the bottom shows just basically from 2011 to 2020. Our data was used in the Edwards study, but theirs extended from 2000 to 2020. But this is the time period where they were seeing significant decreases in butterfly abundance. So this June, John McLaughlin at Western and I will be looking at our data, trying to see if we see the same patterns and if it's continued since 2020. This is just a slide to show you changes, compare abundance for, on the left, it's purplish copper and Western meadow fritillary, two different colors. On the right, it's Mariposa copper and Mormon frit, and the x-axis goes from 2011 to 2024. So the two on the left, those two species were species, or are species, that the Edwards study found was declining both nationally and in the Pacific Northwest. The two on the right are, in their study, were found to be increasing. So we're going to look more at this and see what patterns we see across our sites. And then finally on plant peak flowering, we're just getting this data together, but we're looking at, can we see annual differences in flowering and fruiting times? And do the periods of timing parallel with butterflies? That's a big question with climate change studies. Do some species adapt more quickly or move their locations faster than others? And as Barry mentioned, with pollinators and plants, this is very important. So this is Lupinus latifolius, and it's just on 3 of our sites. So the 3 colors, green, blue, and orange, correspond to 3 different sites. Blue is Mazama, orange is Sunrise, and green is Sauk. Solid lines are 2015, the early snowmelt year, and the dotted lines are 2024, which was a later year. So on the left, it's peak flowering. On the right, it's later senescing. And this is percent of the plants. So you can see it, at all 3 sites, peak flowering in 2015, the solid lines, was much earlier in the summer than 2024. And the same goes with senescing. So these would be flowers in seed, were much earlier on the lupins in 2015 than they were in 2024. This is just comparing Anna's blue abundance on the right, on the 3 sites, with that on the, with the lupins on the left. So lupins are important for blues because it's a host plant, although they also collect nectar from those plants. And you can see that both the plants on the left, the solid lines, are early season for peak flowering, and Anna's blue butterflies were peak early season also in 2015. This slide also shows you how late Mazama Ridge is, or it has been over our years, on both peak flowering and abundance of Anna's blue. This graph compares Erigeron to Erigeron peregrinus and glacialis. I lumped them because on Sauk Mountain we have both, and they're often difficult to distinguish. But same format of the graph. Solid lines are 2015, dotted lines are 2024, and the Mormon frit, which is the early season butterfly, at least on Sauk, is very visible on the right, and much later abundant on Mazama Ridge in 2024. But erigerons are important for butterflies because they're one of the latest flowering species. So you see a lot of butterflies nectaring on erigerons later in the season. And really, none of this would be possible without our volunteers. We're completely volunteer run now since 2019. We've been completely volunteers, and we have some people who have been with us since 2011, some who have been out there for the last 6 to 8 years. And it's great because when you have to learn 56 butterflies, you really need to come back more frequently, both within a season or over several years. So that's all that I have.
You will give people a minute to type in their questions and comments. And just such a delight to see all those butterflies. Summer is coming. Your photos were wonderful.
Oh, thanks.
I'm going to get this. There we go. Okay, so we have, let's see, very fun to see all of your work and data. Regina, thank you, from Barry. So I think we're all enjoying seeing all these summer pictures tonight. We're so close, it's just not here yet, but we can feel it coming. So, and I think some of your volunteers may be out in the audience tonight too. I just see some, yeah. Mm-hmm. It's exciting to think about all those locations that you study and all your sites and the opportunity to contribute to citizen science and be out in those beautiful places and learning about the butterflies. So wonderful work. It's got to be very rewarding. Thank you for sharing that with us. Okay, so I'm going to remind everybody also that we have next Tuesday, April 15th at 7 p.m., our program on rare native plants and related research. And please do go to the WNPS.org website so you can see all the offerings for Native Plant Appreciation Month. There will be a Bio Blitz that's coming on April 20th to 30th. Please read up about them on the website, and we invite you all to participate. Our presenters, too, are invited. So you are welcome to get involved, and I hope you join us and learn more at our website. Thank you for coming, everyone.
Just want to say thank you, Julie, and thank you, Regina and Barry, for a lovely program tonight. This program has been recorded and will be posted on the virtual events page tomorrow evening. So thank you all for joining us and for your participation with Washington Native Plant Society. Please feel free to sign into our umbrella BioBlitz project on iNaturalist. Information is on the Native Plant Appreciation Month page, easily linked from the homepage of WNPS.org. Thank you.
Thank you for mentioning that, Denise. It's an exciting way to involve everybody this season. So please do join us.
We have 8 projects so far, and I welcome individual chapter, hike, individual field trips. Please send me a project name. I'm happy to pull it into our umbrella project. Thank you.
Families, homeschools, school groups, all are invited.
Very good.
Okay.
Very good. Thanks, Julie.
Yep.
Thank you
Good
all
night,
for being
everyone.
here tonight. Good night.
Good night.